Another highly researched hallmark of cellular aging is telomere attrition. But before understanding telomere attrition, you need to know what a telomere even is. So inside your cell, you have chromosome. The chromosome is a linear chromosome. That is, it has DNA in a straight line. And this linear chromosome has two terminal ends. And these two terminal ends are chromosomal caps. These chromosomal caps are referred to as telomere. So one linear chromosome has two terminal ends where two telomeres are present that form the chromosomal caps. And these chromosomal caps have two major functions. Function number one, they prevent chromosomal end sticking. See, this right here is a linear chromosome. At the two terminal ends of the chromosome, there are the two chromosomal caps or telomeres. Due to this chromosomal cap, the end of one chromosome does not stick with the end of another chromosome. So it's preventing the sticking of the ends of chromosome. Second major function, it prevents DNA loss during replication. See, this is what I mean. Inside chromosome, you have linear DNA, and making the terminal part of this linear DNA is the telomere. Now, during replication of the DNA, the terminal part of the DNA does not replicate, and this is referred to as end replication defect. But fortunately, at the terminal part of the DNA, do we have the important coding DNA? No, we don't. We have telomere, which is non-coding part. So with every replication, there is end replication defect, and due to end replication defect, the terminal end or part of telomere does not get replicated. And that is why with every replication, we are losing a part of telomere. And that is why the length of the telomere gets shorter every replication. Again, during DNA replication, the terminal end of the DNA shows a defect in replication, which is referred to as end replication defect. And at the terminal end of the DNA, you have the telomere. Due to end replication defect, part of telomere does not get replicated. Since part of telomere is not getting replicated, so with every cell division, we are losing that small part of telomere. So every replication, we are losing part of telomere again and again and again. That means the length of the telomere, it grows shorter and shorter and shorter with every replication. See, here's the concept. One telomere at the time of birth contains at around 10,000 to 15,000 base pairs. These base pairs are nothing but repetitive DNA sequence. What sequence? T, T, A, Z, Z, Z. So there is a frequent repetition of these 6 base pairs again and again and again, till the number becomes 10,000 to 15,000. This is at the time of birth. So you have T, T, A, Z, 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 T, T, A, Z, 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 T, T, A, Z, 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 again and again and again, till the number is around 10,000 to 15,000 base pairs. But now, the cell goes through cell division. As it goes through cell division, there is DNA replication. During DNA replication, there is end replication defect. Due to end replication defect, part of telomere does not get replicated. As a result, you lose a part of telomere. You're losing some of these base pairs with every replication. And that is why with every replication, see the length of the telomere, it is getting shorter and shorter and shorter and shorter and shorter. Eventually, this cell which I'm talking about, this cell with telomere shortening with every division, this cell eventually loses telomere. There is no longer any telomere. As soon as there is loss of this protective cap, the DNA replication gets arrested. The cell does not divide. Now the cell has become old. Cellular aging has occurred, or cellular senescence has occurred. Since the reason behind cellular senescence is stoppage of replication, we also refer to it as replicative senescence. Okay, cellular senescence is achieved due to arrest in cell replication. So there is a replicative senescence. That is, now the cell will not divide. The cell division has permanently stopped. Okay, but then there is a special enzyme by the name of telomerase. Even though it has A's at the end and it seems as if telomerase is responsible for breaking down telomere, it's actually quite the opposite. Telomerase is RNA dependent DNA polymerase, and the function of telomerase is actually to prevent telomere shortening. Now imagine in your cell there is telomerase activity, unlimited telomerase activity. So in your cell, if there is unlimited telomerase activity, that means telomere length will never become small. The telomere length will remain as it is. With every cell division, there is no change in telomere length. That means the cell is able to show unlimited cell division. That means the cell will never become old. Cellular aging will never occur. That means the cell will become immortal. You will become immortal. So hypothetically, if every cell in your body contains unlimited telomerase activity, you can become immortal. That is why this enzyme is also referred to as immortality enzyme. Okay. In Robbins, a graph has been given in order to compare telomerase activity of 4 different types of cells, and they are the somatic cell, number 2 stem cell, number 3 germ cell, and number 4 cancer cell. So let's compare the telomerase activity. In somatic cell, there is no telomerase activity. That means with every cell division, the telomere length is gonna go down, decrease in telomere length. In stem cell, there is low telomerase activity. So with every cell division, the telomere length will go down, but it will go down slower because the little telomerase activity is preventing the decrease in length. So there is slow decrease. In germ cell, on the other hand, slow decrease. In germ cell, on the other hand, there is adequate telomerase activity. And this telomerase is obviously going to prevent the shortening of telomere length. So there is no decrease in telomere length. In cancer cell, see, cancer cell before was a normal somatic cell or a normal stem cell or a normal germ cell, whatever. But this cancer cell, due to mutation in the normal cell, now has become a cancer cell. Let's say this cancer cell is arising from somatic cell. Okay. Previously, in this somatic cell, there was no telomerase activity. That means telomere length would decrease with every cell division. But now, due to mutation in somatic cell, it has become cancer cell. Once the mutation occurs, once the somatic cell becomes cancer cell, now the previous somatic cell where there was no telomerase activity, now all of a sudden has telomerase activity. That means there is activation of telomerase activity. Telomerase activity gets activated. So before, with every cell division, the telomere length was decreasing. But now after mutation, after telomerase activity has come back into the cancer cell, the decrease in the length stops. Basically, the telomere does not decrease anymore. And you know the relationship between telomere length and cell division, right? Once the telomere length decreases and decreases and decreases and telomere is no longer there, the cell division stops. What is achieved? Replicative senescence is achieved. Why? Because the replication stops permanently. So let's see how soon is the replication senescence achieved in each of these cells. Look at this graph. So in x-axis you have cell division, and in y-axis you have telomere length. So with every cell division in somatic cell, the telomere length is decreasing. Once it becomes zero, once the telomere length becomes zero, the cell division, it stops completely. See, in somatic cell, there is no telomerase activity at all. That is why the cell division has stopped comparatively faster. Cell division has stopped just here. Just here, the cell division has stopped. But now let's compare it with stem cell. In stem cell, there is little telomerase activity. That means with every cell division, yes, the telomere length will decrease, but the decrease will be slower. So with every cell division, there will be slower decrease, and let's see how far will the cell division go. The cell division, let me use white. Cell division goes till here. This is how much more capacity it holds to show cell division. Stem cell. Compare X of somatic cell with Y of stem cell. Y is much longer, means Y is much larger. That means it shows much more number of cell divisions. Why? Because it has little telomerase activity, which makes the decrease in telomerase length slower. Okay, third, let's compare that with germ cell. In germ cell, there is adequate telomerase activity. That means with every cell division, the telomere length is not decreasing. So see, the telomere length, it remains as it is. With every cell division, the telomere length is remaining as it is. So the cell division is still infinite. The cell division goes and telomere length does not decrease. What about cancer cell? I said earlier, cancer cell are formed once mutation occurs in the somatic cell. So previously, with every cell division, the length was decreasing till here. But now at this point, mutation has occurred. Now the somatic cell has become cancer cell. Previously, there was no telomerase activity. Now there is telomerase activity. So now this cancer cell, telomere length will not decrease due to telomere activity. See, there is no decrease. The cell division from this point on, cell division is happening. But telomere length, is it decreasing? No, telomere length is not decreasing. So that's the comparison. Okay, so in order to prove telomere attrition as an important hallmark of cellular aging, an experiment was carried out. So 2 mice were taken. One normal mouse, and another Thanos mouse, not really, genetically modified mouse. The normal mouse had normal length of telomere, normal telomere length. In genetically modified mouse, the telomere length was shortened. So the telomere length has been shortened. So the normal mouse with a normal telomere showed normal lifespan, but the genetically modified mouse with shortened telomere, since the telomere is short, now the number of cell division is obviously reduced because the telomere will disappear faster. So it showed a shortened lifespan as well. It died much earlier.